All right, so look, we're going to have a best paper prize and a best poster prize. And I'm not going to tell you yet who won, but this is part of the solution. This is our voting box for best poster. So I'm going to put it out there on the registration table, and I would like you guys to put your vote in this box with the number of the poster that you appreciated the most. All right, and then we will take a random slip from the box, and that's the winner. No, we will look at the poster with the most votes, and that's the winner. All right, it will all be certified by me. All right, so I'll put it out there. And then Anna wants to say something about the certificates.
Yeah, many of you will need a certificate of attendance, so please write to info.csnnetwork@upf.edu, specify your name and your institution, and I will be sending you a certificate. Or if you have any other question about a receipt, I will be happy to answer you, and by tomorrow you will have your information. Thank you.
All right, so our next speaker is Takuya Umedachi, who will talk about a true slime mold-inspired fluid-filled robot exhibiting versatile behavior.
Thank you, Chairman. My name is Takuya Umedachi from Hiroshima University. Today, I will talk about the topic entitled True Slime Mold-inspired fluid-filled robot exhibiting versatile behaviors. So this is the table of the contents. First, I will talk about the background of the study and the purpose. And as a proposed model, I will explain real physical robot and the simulation of a mobile robot. Then I will conclude this presentation and talk about future work if we have enough time. Now, let me begin with the background of this study. As you can see, these are movies. Animals, even primitive species, exhibit qualitatively different versatile behaviors and spontaneously switch between these behaviors according to the situation encountered. Based on this behavior diversity, animals are able to generate adaptive behaviors. Now, the question arises, how should we design this behavior diversity for artificial system? And we believe that this behavior diversity should not be random, but should be grounded for their own morphology and should be grounded for their dynamically changing environment. Now, in order to achieve the answer, let's look at the animal kingdom. As you can see these pictures, there's a lot of species and more behaviors. In order to achieve the design principle of behavior diversity, we took a back to basics approach. More specifically, we have focused on a very primitive living organism that is a true slime mold. True slime mold is one giant cell organism that exhibits amoeboid locomotion by generating thickness oscillation, as you can see this movie. It does not possess brain or nervous system, but it exhibits amazingly adaptive, versatile behaviors, such as when the true slime mold cannot find any attractant, it moves around the environment to search attractant. In this condition, the true slime will generate a spiral wave. The colors describe the thickness of the true slime mold. But once it finds the attractant, it moves toward the attractant. In this situation, true slime will generate a traveling wave. So based on this, we can say that true slime mold is able to generate adaptive, versatile behaviors without brain or nervous system. But the question is, how is it possible? There is a key experiment to understand such adaptive versatile behaviors of true slime mold, which was done by Takamatsu et al. She made a physically coupled oscillator system with true slime mold, and she observed the oscillatory pattern with thickness. This is a experimental result. The colors describe the thickness variation. This one shows the rotation mode. As you can see this, the red part moves rotationally. And she also observed the partial anti-phase mode. This one and this one oscillating anti-phase. And the last one is the partial in-phase mode. The bottom one's oscillating in phase, and the top one are oscillating anti-phase compared to the bottom ones. And so, as you can see, this result, the true slime mold can generate versatile oscillatory patterns. And more interestingly, it exhibits a spontaneous switching between these oscillatory modes. And using this behavior diversity, the true slime mold can generate adaptive behaviors according to the situation encountered. So, based on these biological findings, we can say that the true slime mold is one of the most primitive form of intelligence concerning versatility. So, the purpose of this study is to understand this primitive form of intelligence by constructive approach. In particular, by building a model to generate such versatile behaviors, and by implementing the model into a real physical robot, we reproduce this behavior diversity which can be observed in the true slime mold. And we believe that without understanding this primitive form of intelligence, we cannot understand any such intelligence of any higher form of animals, we believe. Now, in order to build such a robot, let's look at the detail of the true slime mold. The true slime mold mainly consists of two parts, elastic outer skin made from gel structure and protoplasm inside the elastic outer skin made from zoll. And by this part contract rhythmically, which leads to a protoplasmic streaming. And one of the most important factors that allows the whole system to generate a versatile behavior is physical communication between the mechanosensory oscillators, we believe. There is three reasons to think so. First, it uses purely decentralized system, which consists of a coupled biochemical oscillator similar to CPZs. And the second, there is a long-distance physical interaction between the oscillators due to the conserved quantity. That is mass of protoplasm. So inside outer skin, there is a protoplasm. Due to this mechanical constraint, when one oscillator expands, the rest of them automatically shrinks. So all oscillators are connected with this long-distance physical interaction. And the third and finally, there is a phase modification mechanism exploiting mechanosensory information, which was reported by Yoshiyama et al. And he reported that the oscillator can feel the force from the protoplasm and can modify its own rhythm to reduce the force from the protoplasm. So in sum, the true slime mold can exhibit versatile behaviors by fully exploiting the physical communication or morphological computation. Now, I'd like to move on to the real physical robot. Before that, I would like to explain the basic concept of the robot. The robot consists of several modules, and this is one module. And the module consists of two air cylinders connected with the tubes. And this, each air cylinder are pushed or pulled by the soft actuator. And the soft actuators are controlled by the phase oscillator. And please note that these oscillators are decoupled, not communicate electrically, but only interact physically. I mean the protoplasm inside the air. Protoplasm inside the cylinder. So there is a the inside cylinder there is a cylinder protoplasm. So if you push one side, the other side moving up immediately. So the system has a physical interaction between stemming from the air. I mean protoplasm. So this is the real physical robot, one module. As I mentioned before, there is a cylinder and connected with the tubes. And to push or pull the protoplasm inside the cylinder, we implemented the sub motor for each cylinder. So by rotating this part by sub motor, we can move up or down, push or pull the protoplasm inside the cylinder. And for visualizing the volume variation, when we put LED, and when volume increases, LED turns on, and when volume decreases, LED turns off, as you can see this. And please note that there is an elastic band between the sub motor and the cylinder rod. As you can see, the target value, I mean the sub motor angle, and the actual angle is there is a gap between them. So due to this soft mechanism, there is always discrepancy between the target value, sub motor angle, and the actual angle. And this actual angle can be detected from the potentiometer we implemented. Now I'd like to move on to how to design control system. In order to reproduce the oscillatory pattern of the truth line mode, we use phase oscillator system, as you can see this equation. And according to the phase of the oscillator, we change control the target angle, sub-motor angle. And as a phase modification mechanism, I would like to explain more detail about this phase modification mechanism. And as a mechanosensory information, we use a torque, which can generated that because due to the softness of the elastic band. We can sense the actual angle by the potentiometer, and we can get the information, target angle, and we can calculate the torque as a mechanosensory information. And based on this torque, we can design discrepancy function. Discrepancy function is a function that detects the discrepancy between the target value and actual value. And we design this discrepancy function so that when absolute value of the torque increase, this function value increase. And based on this discrepancy function, we can design phase modification mechanism so as to reduce its own discrepancy. So the equation is very simple: phase oscillators, intrinsic frequency, and phase modification mechanism. So the oscillator rotate, but feel the force from the protoplasm. They modify their own rhythm to reduce its own discrepancy. Now I'd like to move on to the experimental result with one module. The initial setup, we set the two-phase oscillator in phase condition. And please note that each oscillator are decoupled. So in phase, and pulling the protoplasm inside each cylinder. But due to the second term, phase modification mechanism, that each oscillator modifies its own rhythm to reduce its own discrepancy function. So you can get that phase, they can get anti-phase oscillation. So let's see what is happening inside. Horizontal axis is the time, and the vertical axis is a phase of these graphs plot. And horizontal axis is time, vertical axis is a discrepancy, a total discrepancy. In the beginning, in phase, pushing or pulling the protoplasm, so huge amount of a discrepancy function, but they try to reduce its own discrepancy function, and they can get anti-phase oscillation as a result of just reducing its own discrepancy. And this experiment demonstrates that phase modification occurred so as to reduce its own discrepancy function. And this system is this phase modification occurred in fully decentralized manner. Now, what will happen when we connect the two modules? And we put the air regulator to tune the protoplasmic conductance, I mean air conductance between the modules. And in this experiment, we changed the protoplasmic conductance from high to low condition. So in the beginning, it's easy to exchange protoplasm between the modules. But suddenly, it becomes difficult to exchange protoplasm between the modules. So in the beginning, anti-phase oscillation we set as an initial condition. As you can see, they generate anti-phase oscillations between the module, but situation changed. It becomes difficult to exchange protoplasm, so they can get anti-phase oscillation inside modules. So let's see what is happening inside. Horizontal axis is the time, vertical axis is this plot is a total discrepancy, and these plots are describing the volume of for each modules. And in the beginning, they are exchanging protoplasm. As you can see, the volume is oscillating anti-phase condition, but situation suddenly changed. It becomes difficult to exchange protoplasm, so high value of the total amount of discrepancy, but they have to reduce its own discrepancy, decentralized way, and they can get anti-phase oscillation inside the module. So this experiment demonstrates that the robot behavior changes based on the morphological condition or constraint. And we don't change any parameter during the experiment. And let's see what is happening when we connect the three modules. And this is our experimental result. We observed 4 oscillatory patterns without any change in the parameters. And these plot data were achieved by one consecutive experiment run. Horizontal axis is the time, vertical axis is the volume. And as you can see, the volume peak moving from 0 to 1, 0 to 1. Let's say, so this is a rotation mode. As you can see this, please pay attention at the LED turns on, turns on, turns on, turns on. So that volume moving rotationally. And we also observe the partial in-phase mode. The bottom oscillators are oscillating in phase condition, and the top one oscillating anti-phase compared to the bottom ones. And we also observe the partial anti-phase mode. Bottoms oscillator oscillating anti-phase, and the top one generating anti-phase oscillation inside the module, so they don't require any they don't require protoplasmic exchange between the modules. And we also confirmed that intra-oscillation mode, in this mode, in each module, they are generating anti-phase condition, so they don't require protoplasmic exchange. And more interestingly, we observed transition between all oscillatory mode, as you can see this. Now, I'd like to move on to the mobile robot with the same mechanism. That was the basic concept of the real physical robot, and we rearrange the air cylinder positions like this, and we put the friction control unit on the edge of the soft actuator, as you can see this, in order to generate locomotion. And we add module like this, and connect it with the tube in order to generate protoplasmic streaming between the modules. And here is the whole arrangement of the mobile robot. So let's see let's see how robot moves. Without, in the absence of the attractant, exploiting the transition between the all four oscillatory mode, the robot moves around the environment. This is the whole trajectory of the robot. As you can see, the robot moving around the environment. But once the robot finds the attractant, we change just softness of the soft actuators like this. When the attractant side, the soft actuator becomes softer than the other part. And this generates taxis locomotion, exploitation behaviors. So as you can see this. So this is a whole trajectory of the mobile robot. As you can see that robot can approach the attractant. Now I would like to conclude this presentation. We reproduce versatile oscillatory patterns and spontaneous transition between them by fully exploiting the morphological computation. And we don't design complex diffusion network, and we don't design complex oscillatory dynamics, just phase oscillator interacted with the using the phase oscillators and using the interaction between the oscillator with the using physics, but we can generate versatile oscillatory patterns. And we also demonstrate that the mechanism can be applicable for the mobile robots to generate exploration and exploitation behaviors in the simulation. As a future work, we will investigate, we have to investigate the essential mechanism that induce such oscillatory patterns, and we have to build a real physical mobile robot to generate exploration and exploitation behavior. Thank you.
Thank you very much. Great. Any questions? Barry.
A very, very interesting demonstration of the computational morphology going on there. Can you dynamically and continuously adjust the elasticity of that coupling to explore the range of possible modes? Because you said you got switching between modes. That was spontaneous, is that correct?
Yes.
Okay. And can you predict when that's going to happen and adjust the stiffnesses of the elastic coupling to control that?
You mean the structure of the transition of the oscillatory mode?
Yeah. So if you had a way to continuously change the elastic band, can you then create a range of controls? Yeah.
Yeah, that was easy one, because we do a lot of simulations, and this is another simulation result. But please imagine that if you this one is hard, middle, soft, we can easily generate oscillatory mode. So optimizing the softness, by optimizing the softness, you can generate the oscillatory mode purposely. But what we want to do is I think that life is like the, when you put on the robot on the floor without any control, they don't move, but the, living system moving around without any, attractant or stimulus. So we would like to reproduce that kind of, behaviors, and we think that this is a key, big difference between the robot and real life. So yeah.
There was another question.
There's a fellow named John Trotter at University of New Mexico that's demonstrated that there are tissues in sea cucumbers that undergo a gel-sol conversion under neural control. What is it, do you think, about the environment that causes the gel-sol conversions that characterize these movements?
I can understand your question.
What in the dictyostelium, what is it that is causing the gel-sol conversions that cause the movements?
You mean in the real true
Yeah, in the real beast. Yeah.
It's like, it's a big mystery. So on the front area, the layer is so thin, and to push the tail, push the protoplasm, and the front area so thin, so the protoplasm, getting out, and the surface of the protoplasm become new soft outer skin. That was the case of the true slime mode. Yeah. So does that answer your question?
Well, I was just wondering, I mean, if they're going to be attracted to an attractant, you would think that that would cause an elongation in that direction. And I'm just wondering if the nature of the signal is understood.
Yeah, imitating all things is impossible.
Control signal. What's the signal in the environment that would control this change in conformation of the slime mold?
Yeah.
What's a controlling signal?
Okay, I understand the question. The true slime, true slime can sense the the environment just touching. So if the true slime can touch some kind of nutritions, that part becomes softer than the other part.
Yeah.
So the true slime was just gathering, just one single cell organism, but the whole volume coming to the, nutrition area.
Yeah.
So does that answer your question?
Okay. Yeah.
Other questions? Okay, then thank you very much for your really nice presentation. Then the next speaker can set up, which is a talk on the emergence of action sequences from spatial attention, insight from rodent-like robots, by Ben Mitchinson, but he has sent his assistant, I understand. So Tony will stand in. But now, Joe, you were saying something about the sea cucumber being under neural control for these transitions?
It's a gel-sol transition.
Can you
explain how that works? No, not quickly. I know of the phenomenon. I don't know the exact mechanism. But there are motor neurons, and the motor neurons impinge on this connective tissue, and that causes the connective tissue to undergo from a gel configuration to a sol configuration, and that's how the sea cucumber does its shape change.
There's still a membrane around it.
Yeah. So it's physically delimited connective tissue, but it's the neural input that actually causes a gel-sol conversion. Yeah, it's one of the craziest actuators I've ever seen.
Great, thank you. All right, Tony.
Okay, we're ready. Yeah. So I think Mitch is here, and he's been giving the demo the last 3 days, and is available to answer questions. And so Mitch is the lead author, and Martin, who's also here and has been demonstrating Shrubot, is the lead developer of the hardware that I'm going to talk about. And then myself and Tony Pipe at Bristol Robotics have been the sort of chief investigators for this project, and it's all funded by Future Emerging Technologies, part of the European Commission, a project called Biotact, which sadly is just ending. This is our model system. So we study mammals. There's fantastic work on invertebrates in this meeting. I'd like to see more work on mammals coming through. There are people that are working on mammal-like robots. I think that there's a lot fewer of us than there are actually people building invertebrate robots, unless, of course, you count humans, and then we're all outnumbered by the humanoid people. But I think that this is a fantastic model system for us to look at. Let me just show this again. This is real time, and this animal is exploring in the daylight, but it really wouldn't make any difference if it was dark, because he uses his whiskers more than he uses his eyes to explore the world and to move around. And when people first started looking at the movement of the whiskers, they thought, oh, stereotype pattern is moving them back and forth and whacking them into things. It turns out it's not at all like that. If we play the movie Slow Speed, then what the whiskers are doing changes with what the animal is doing. When he's running along, the whiskers aren't flying back and forth. They're generally pushed forward, trying to anticipate things that he might bump into. When he reaches over an edge here, again, there's big sweeps, but now he's reaching to see if he can find something that's in the space. When he encounters an edge at an oblique angle, you see the whiskers tilt with the shape of the objects he's investigating. Here you see really quite precise and accurate contacts of the whiskers with an object. So I would contend that actually this animal is using its whiskers the way that we use our fingertips. And actually we're the only mammalian species not to have whiskers at some point in our lives. And the primates gradually stopped using their whiskers as they developed these things, which are really whiskers on sticks. So this system can tell us an awful lot about how the brain controls movement. It's a simple system in which there are several degrees of freedom in the whiskers, but we can measure them, we know about the actuation, and we can learn a lot, I'm going to tell you today, about attention in brains through studying this system. So the paper that we've put into the proceedings is really about the question of the decomposition of control, and it's something I think we've touched on in our discussions, is how you decompose the control of robots and what we can learn about the decomposition of control from looking at animals. This is from the ethology literature. This is by a very famous ethologist called Behrens, and this is a study of the herring gull that he did. And he proposed that you could break down the behavior of the herring gull into these behavioral systems: settling, building, locomotion, trimming, bathing, and each of those could be further broken down into their components, which some people have called fixed action patterns. So that the decomposition control is about deciding which of these general behaviors you want to do at any time, and then getting the right sequence of the components of that general behavior. Now, this has been quite inspirational in the robotics literature. Of course, Rodney Brooks developed the subsumption architecture based on this principle of behavioral decomposition of control. This is a version of a behavioral decomposition that we've implemented in several robots. It's not exactly the subsumption architecture, because what we generally have done is taken behaviors, or fixed action patterns if you like, and put them into a selection mechanism, and the selection mechanism decided what to do at any time, and then it's inhibited or removed the inhibition from the behavior, and then you get movement. And we have published a lot of papers arguing that there are these kinds of central selection mechanism in a part of the brain called the basal ganglia. Now, if you've had a chance to see Shrewbot, this is the kind of thing that Shrewbot does, sort of investigating a tube here. And what I want you to pay attention to just now is just where Shrewbot is orienting its snout. And the control system that is moving Shrewbot around in this case is based on this behavioral decomposition. We have 3 behaviors, explore, orient, and what's B, Mitch? Back off, thanks. And this is an example where we've got explore is the smooth line. This is orient. And then we have the other back off up here, and then we have some more exploring. And this is a traditional way of displaying this kind of decomposition of control over time, an ethogram that the ethologists developed, and we can easily apply it to robots. And in this case, we can actually take from the control program for Shrewbot exactly what it was doing and generate the ethogram as we go along. Oops. Sorry. But there's another way to think about the decomposition of control. And many people, when they thought about how attention is controlled in mammals, have argued that what we do is we compute a saliency map of whereabouts in space we want to put our attention. And then we select points within the saliency map of high salience, and we go towards those. So this is from the visual attention literature, where there have been many models. Itty and Koch is one of the most famous ones, and this is an example of a saliency map computed from this picture, a saliency map computed from this picture, and then you take the saliency map and you use that as the input to a selection mechanism that selects a peak of high salience and foveates your attention, your gaze towards that peak. And the hypothesis is that we can use human eye movements, saccadic eye movements, to find out about where our spatial attention is, because our fovea is really quite small, and we move our fovea very rapidly to different points in space that presumably we're interested in because they're highly salient. So this is a very famous picture and an example of eye movements of somebody scanning it. And of course, the eyes and the mouth and the nose turn out to be the points of interest that the eyes keep returning to. So this is a general model of how you might implement a saliency map type model for attention. So we are going to think about mapping onto this brain if we have time at the end, but we imagine that there are some reflex arcs. Not everything is going through the saliency map, but we also have a map of the local space, and we have a system that can decide whereabouts in local space the most salient positions are, and can move the eyes or whatever sensor system you're interested in, and direct them at that position in space. So, and there may be some more abstract mechanisms, perhaps cortical mechanisms that are also influencing this, but the key point is that somewhere there might be a map of space where you calculate the overall salience of different positions. Here's Shrewbot running with a saliency map model for attention. Now, his behavior looks slightly different. His whiskers are actually moving more slowly, but not because we're using this model. He's also a bit more sort of punctate and discrete in his movements. But otherwise, if you look at his behavior, and here Mitch has coded the video into different categories of behavior based on what we did with our behavioral decomposition, so exploring and orienting and backing off, then you can similarly decode the behavior of the robot. So at least in observational terms, it's very difficult to know which is the underlying mechanism. Is it a behavioral decomposition, or is there a saliency map which is deciding where attention is going, and that then is driving attention? And I think we've discussed this a little bit in the meeting, this issue, and of course it goes right the way through science, that we have to understand the mechanisms. You can't just look at the behavior, you have to look at the mechanisms and the behavior together, and then you can generally get a much better idea of how control is happening. So I want to talk about the whiskers. This wasn't really in our paper, but the larger model which we're trying to publish this year is about how you don't just control the positioning of the head, where you're looking, if you're a rat, you're also controlling the whiskers in a really quite precise way. I want to show you some data that we've collected over the years showing that rats really do accurately control their whiskers. This is something that we call rapid cessation of protraction, which is the idea that rats have a light touch. When the whiskers move against something, they don't bend them hard against the surface. They touch and then come off again. It's the same if you're using your fingertips. If you know where the surface is, then you'll touch it lightly. You don't want to damage your fingers. And here is a rat. You'll see the whiskers on both sides moving together. There's a contact there. Those whiskers start to retract while those ones are still going forward. So there's a light touch there, and then the whiskers retract that side, the other one's still going forward, and then after a couple more cycles, they go back in to synchrony. Here's another example of what we call an active sensing strategy. So this is a young rat, I think it's 16 days old, and what it's doing is it's turning, and as you're turning your head, obviously you look in the direction that you're turning, so your eyes will be looking this way, anticipating what's in the direction of the turn. And he's doing something very similar with his whiskers. As he's turning his head, the whiskers are asymmetric round the head, so that they're looking in the direction he's going, anticipating the space that he's going to be moving into. We call that head-turning asymmetry. It was described by Blythe Towle and Mitra Hartman. Here's something else that rats do when they come up to a surface. So the whiskers are generally quite spread out. When it comes up to a surface and starts to investigate, the whiskers come together. And naturally, that results in more contacts on the whiskers. So when he's exploring in space, they're spread out. When he comes up to a surface that's interesting, brings them together and touches them against the wall. And this is another form of asymmetry. So we had asymmetry when the head is turning, and there's also asymmetry when he contacts an object. So you'll see this is actually a wild rat. We wanted to make sure it wasn't just lab rats that did this. He's got slightly bushier whiskers, but otherwise behavior is much the same. You see the symmetric movements, and then when he comes up to the block and moves along one side of it, you see it become really quite asymmetric, with these whiskers moving a lot less, and those ones a lot more. So these ones are lightly touching, these ones are reaching round, trying to find the block. That's just another example. We'll move on. And I mentioned this before, that there's also a kind of look ahead, that when the rat is running, rather than whisking and exploring, the faster he goes, the more he'll push the whiskers forward. We call that the set point. The whiskers get pushed forward, and they go from being big amplitude sweeps to small amplitude sweeps, because he's interested in what's in front of him or what he might bump into. You can also see that if he's running, he lifts his head up, the body posture changes, his attention changes to be directed ahead of him. So we can summarize a lot of these different observations with what we call minimal impingement, maximal contact. The idea being that the rat is trying to maximize the number of whiskers that contact surfaces of interest, at the same time minimize the amount that the whiskers bend against those surfaces, minimally impinge against the surfaces. So taking that hypothesis, what we are arguing now is that maybe what we can do with this observations is really say something about the spatial attention of the rat, that we can look at the vibrisal behavior, and there's many different degrees of freedom for controlling vibrisal behavior, and we can really know something about what it is in space that the rat is interested in, because we can see how he's moving his whiskers, where his attention is going. And the hypothesis we're investigating then is that there might be a saliency map, which is he's using not just to position his gaze, but also to move the whiskers and position them precisely in areas of the space that he wants to know more about. So when we started to implement this, we thought, well, maybe we could just do it with reflex loops. How much time have I got?
10 minutes.
Oh, good, okay. So maybe we could just have a bunch of reflex loops, and reflex loops could do things like the rapid cessation of protraction, touch something, you have a reflex that automatically makes the whiskers stop protracting and begin to retract. We could do head-turning asymmetry. We could imagine that as a sort of vestibular vibrisal reflex loop. We could do that for look ahead. all of these things, we could imagine having low-level systems which take information from the whiskers, feedback onto the motor pattern generators, and just adjust the gains. And if we do that, we can get something that looks a little bit like some of the whisker patterns that we see in the animals. And we've done that on ShrewBot and our other robots. This is a previous robot called ScratchBot, where we implemented a reflex version of various of these active sensing strategies, including the rapid cessation of protraction. And the problem was to make them all work at the same time. So you could tune it so that one of the reflexes would work quite well in some situations. But to make all of these reflexes work in a nice way on the physical platform in a way that's insensitive to where the obstacles are turned out to be very difficult, and there were a lot of parameters to tweak. So we wondered whether really that was the way that the rat was solving the problem. Does he have a whole bunch of reflex loops that adjust the position of the whiskers to give him something like minimal impingement, maximum contact? Or is it possible, that there is a bit more cognition in the rat brain, that he's computing a saliency map, perhaps in the way that we do, that he knows where his whiskers are, and he's positioning them precisely in space where he wants to get more information. So that's what we've been investigating. We've been thinking about building this saliency map on the basis that he's using minimal impingement, maximal contact control, and that he's using a saliency map to work out where in space he wants to investigate a region, not just a point, because unlike the fovea, which just looks at a point, the whiskers can pick out a region, and then it can be wider or smaller depending on how you modify the spread of the whiskers. It can be directly in front of you and orthogonal, or it could be slanted depending on how you control the symmetry. And that region of interest is then driving where the whiskers are moved and how much each whisker is moved. So this is our model. I'll skip to the next slide here. So we have an oscillator generating whisker movement. We have a spatial attention map. We have a gain which is being put onto the oscillator so that we can change the movements of the whiskers. And then we also have a signal from the spatial map as to the point in space you want to foveate. One of the things that we need to have in our spatial attention model is inhibition of return, because you don't want to keep going back to a place you've just investigated. And inhibition return is something that people have studied a lot in the human visual system. We definitely have inhibition of return. It actually turns out to be complicated to understand exactly how to implement it properly, but we need that in our system too, so he doesn't keep going back to the same point in space he was at a few seconds earlier. So, yeah, very quickly. So he needs a map of the world. He needs to know where he's been before. That gives you the inhibition return. You don't want to go back somewhere I've explored recently. More generally, he wants to inhibit bits of space that, for instance, are inside the head. I don't want to go and explore the area of space that I'm currently occupying. So the three components inside of the snout, favoring the near the snout, inhibition of return. And we take the maximum across these three components. So this is the positive aspect of salience, and these are the negative aspects of salience. And we compute a region of interest in space, and we use that to control positioning of the whiskers. And quickly show you the results. So this is how we compare to the biology. So this is all simulation results, not robotics, but we have it now working in robots. And this is the performance of a 2D model controlled by this saliency map system. And these are the data from Towle and Hartman from the original paper, and some work by ourselves also finding this head-turning asymmetry in rats. This is the contact-induced asymmetry where the rat encounters an object asymmetrically, and it moves its whiskers to explore the different aspects of the object, protracting further for the whiskers that are further away and protracting less for the whiskers that are nearby. I don't have time to explain these plots, but just to notice that the model is generating the same kind of contact-induced asymmetry as we see in the biology, and we've seen that now in several species we've looked at. this is the control of spread. And again, so when he gets close to an object, you can control the whiskers, bring them together, and that way make more contacts with the object. Again, the biology has the model has a qualitative fit to the biology. So this is Shrubot doing some exploring of, I think, Martin, is that your hand? And so we're at the stage now of quantifying to what extent this is an effective strategy and how much it would improve on the reflex loops model that we had earlier in terms of giving us good contacts. What we want is many contacts. We also want the contacts to be light, sort of within a narrow dynamic range, so that within that range we can have really good sensitivity. How much time? Oh, good. Okay. So I want to talk a little bit about where this thing might be in the brain. So this is a very rough picture of the architecture of the mammalian brain, particularly drawn for, obviously, the vibrissae system. But you could draw a similar diagram for every vertebrate. You could also describe for the human and the primate visual system the same kinds of loops. So there's a brainstem loop, a midbrain loop, and a loop that goes through cortical areas. There's a structure called the basal ganglia, which we think is involved in selection, action selection. There's a structure called the cerebellum, of course, which is involved in modulating movement and often making it more precise. We have various models of the role of the cerebellum in the whisker system. And there's a structure called the superior colliculus, which is viewed by many people as the gaze control system of the mammalian brain. So, and this is an example of, I think, what is the current view of many people in the community, that the colliculus, the superior colliculus, usually studied in the context of vision, but of course it's multimodal. Activity in movement-related cells in the colliculus represents a general command for a change in the direction of gaze, and not a specific command for moving either the eyes or the head. So the idea is that the colliculus is telling you the place in space that you're interested in, and then the brainstem has the job of actually getting the eyes and the head in the right place so that you can look at it. If we look at the colliculus, and this is a work from Drager and Hubel, 1976, we see that there isn't just a visual map, there's also a map for the vibrissae, which is overlaid on the visual map. And actually the whiskers, the regions for each whisker are blurred over the visual map, and that's, of course, because the whiskers move in space. So they're not stationary, they're moving, and they're sweeping across the space. And one of our hypotheses is that if you were to record in some of these collicular cells, then you would see that they would be sensitive to the whiskers in a particular position in their sweep. And that's what they're coding for coding for a point in space, and then when the whisker reaches that point in space, they would become active. And activity in the colliculus, it gets a very rapid signal from the whiskers, about 5 milliseconds, and you have a response in the colliculus, a strong response. If you stimulate in the colliculus, you can get changes in the amplitude of the whisker movements. So the control that we need to have to do the asymmetry, we can get that in the colliculus. It looks as if the level of activity in the colliculus controls the extent of the sweep. So we are proposing what you might call the whisker displacement hypothesis. Activity in the deep layer of superior colliculus could provide a whisker displacement command for both the head and, I should say, whisker movements, so that the deep colliculus is coding whereabouts in space it wants the head to go and then the whiskers to follow. And I just want to show you another vibrissae robot that we have. This one is attached to a robot arm. I haven't had time to mention that one of the things that's interesting about this system, and I think it would be great to explore it, there's a soft, deformable thing called the mystacial pad that the whiskers sit in. And this system is, I think, what we might call a muscular hydrostat. There's about 4 different groups of muscles, including each whisker has an individual muscle. And what we're trying to do is get a bit closer. It's not quite a soft robot, but you now have actuators, so we can move the rows of whiskers around, and that gives us another degree of freedom for positioning them. Let me just put the conclusions up. This is a great picture that you can see in the Miro Gallery in Barcelona if you have time today. And I'm delighted that Miro puts whiskers into his pictures. Thank you very much.
All right, that's great. Question.
I've noticed that the tip of the nose, when it makes contact, seems to be associated with the transitions. Is that sort of proboscoid or what? You know, the actual tip, the fleshy part of the nose.
Yeah, well, it's very sensitive.
Yeah.
So what do you think it does?
Do you think it maybe play a role in some of the postural transitions of the whole head?
Well, I think what he's doing is not just exploring with the whiskers, he's exploring with all the sensors in that area of the face. So obviously sniffing is synchronized with whisking and probably controlled in this active way. There's a recent hypothesis that actually the pattern generator for sniffing is driving the pattern generators for whisking, although in a slightly decoupled way, which is really interesting. And then he's also got all the sort of chemosensory systems around the mouth and the nose, and so there's a lot going on there. Also, there's lots of short whiskers, the microvibrissae, and they're really the fovea of this system. So he's putting the microvibrissae onto objects that he's interested in. And you can't see them in these videos, but you have other videos where you can really see close up the microvibrissae dabbing against surfaces.
Yeah, first of all, very interesting talk. I was curious to learn a little more about the integration of vision in whisking. I don't know much about rats. I know in cats they have rather poor near-distance vision. So that means that if there's a mechanism to integrate vision with whisking, you would anticipate that there will be some kind of convolution in an optical flow kind of way, that's something that you've seen before and you're moving at a certain speed, you anticipate that you're going to pick it up from whiskers later. And are you seeing that in rats, that there's this sort of time delay way of doing the integration?
I mean, what we tend to do is to try and control for vision by either filming infrared in the dark, or we have some animals with a genetic mutation, which means they're actually blind by the time we're studying them. We do film sighted animals. Generally, they look to be doing much the same thing in terms of their whisking behavior as the unsighted ones. But we haven't quantified this. I would say maybe the unsighted ones do investigate for the whiskers a bit longer, and you would expect that the sighted ones would have more anticipation, but we haven't quantified it. Certainly the colliculus, the maps lie over each other, but also the vision system is really looking up, and it's a defense system to alert you to, sort of hazardous objects like birds that might be flying overhead. The whiskers look down and they explore the space, so they're more about approach, and the vision system is more about avoidance.
Hi, nice talk. So I have a question and comment. Did I understand correctly that you're building a saliency map out of the active process of whiskering around?
Yeah, so the key input to the saliency map is what I've contacted recently. So if I contact something new, that's then, if you take the analogy with the visual saliency map, that becomes a hotspot in my tactile saliency map, that I've contacted a space and I've not previously
Okay,
explored
so there's something about saliency maps. I mean, why do we have saliency maps in vision? And that is, there's an economy principle. You have all these things out there, and you don't have time to really go and see and figure out what they are. But in this case, you're already spending the time to really go and figure out what they are. So I'm not sure this is really quite saliency in the traditional way. And the second comment, I guess, is that it would be interesting to see if anybody here is a bad physiologist. I mean, I would think an animal that has a sensorium with active senses is the bad, do the bat have a saliency, build saliency maps of the environment acoustically, you know, so anyway.
I've no idea. I would think that they might. It's not a very rich representation. It's a representation of where contacts have been made, and you need some kind of spatial map to say where I've been before so you can get inhibition of return. And we're not saying all of this maps onto the colliculus. The idea is that the colliculus would be the integrator where this information comes in, and that's where you would have hotspots for salience. But obviously, actually, cortex is going to be involved. And elsewhere in our group, we're also looking at saliency maps for active vision. And the general hypothesis that in the primate visual system and in the rat vibrissae system, there is going to be the same underlying architecture for controlling attention. And what we're claiming is that perhaps rather than deciding what behavior you're going to do first and then thinking about decomposing that the rat decides whereabouts in space its interest is, and then perhaps later you decide what behavior you're going to do with respect to that point in space. And that the whiskers can really show us where this point of space is in some detail.
Last question, Louis?
Thank you. Very interesting and comprehensive talk, Tony. I think this is very related to the DAC structure that Cesar presented yesterday about reactive and adaptive. And then I'm interested in, well, you have said like a highest layer in which you said something like memory. Mm-hmm. Okay. What I am interested here is, apart from this saliency map, when you are supposed to do the difference between the exploration and exploitation, more or less. If we, for example, try to do an adaptive and learning behavior, and if this could be related, you said a saliency map. At the same time, you have said that basal ganglia could be related to action selection. So that's my question. If you set here some action hierarchy to select in order to set the sequence of actions? Or?
I mean, I think that one of the things I didn't have time to talk very much about is that obviously attention, some of the things that derive attention are external and many of them are internal. So there's the overt cues for attention, and then there's covert stuff which we have to guess about or measure in different ways. And so the hypothesis is that the colliculus is controlling where you want to direct your attention, but it's going to get inputs from many other parts of the brain, and actually the basal ganglia may be the structure that is deciding which is the hotspot in the colliculus which I should attend to now. So in our larger model, we have the basal ganglia actually looking at the saliency map and saying this is the bit we want to attend to, and cortex is going to be involved in things like inhibition, return, and memory, definitely, and there's very strong links between barrel cortex and colliculus that we've still yet to build into our model, but that's where we're going. We're trying to head towards a model with cortex, colliculus, basal ganglia, cerebellum, all working together to drive attention and control the whiskers.
Very good, thank you. Thank you, Tony. Just one thing. I mean, as a Dutch patriot, I'm a bit surprised that you do not show any reproductions of the marvelous work of the masters of Dutch painting of the 16th and 17th century that present lots of organisms with loads of facial hair.
All right. Okay.
So why do you have to jump to Miro? It's a bit
we're in Barcelona.
Right, okay, good. All right, Mark, you're on. Okay, so our next speaker is Mark Kotsky, and he's going to talk about a jumping robot with a tunable suspension based on artificial muscles. And for those of you who are escaping earlier, don't forget to drop your vote in the voting box. Otherwise, I will keep the best poster prize for myself. And I deserve it. Andreas made an important point. I don't really believe this semi-map analogy works, because
All right, well, good morning, everyone, or I guess just about noon. I'm going to talk about a fairly focused project that we've been working on for a couple of years as part of the broader picture of building bio-inspired robots. And although many of the talks in the last day or two have really been focusing on mechanisms of control, reflexes, very interesting talks about neural systems, learning, and so on, as a mechanical engineer, I'm equally interested in how materials and structures contribute to the performance of biological systems, to improve robustness or to simplify control. So the specific motivation for what I'm doing here is that subsequent to our work on climbing robots, we've started to build flying machines that autonomously fly and land on walls using actually the same attachment mechanisms that we were using earlier for SpinyBot. And this little airplane is using a laser rangefinder, and as it flies at about 10 meters per second, and when it's about 6 meters away from the wall, there's an inertial measurement unit and an autopilot on board, and it executes a controlled pitch-up maneuver to lose a lot of its speed and present feet for landing. And as you can see, when you have it tuned up, it works fairly well, but it requires a fairly precise and rapid maneuver. And that's mainly because we're using a completely passive suspension system here. It's a very nonlinear, very damp system. And the consequence of that is that we need to be pretty careful about how we execute that maneuver. And in contrast, there's this nice video here from Andrew Spence with the Colugos, and you would see pretty much the same thing in the US with the flying squirrel. It really is able to land under a wide range of conditions, and it's doing some very interesting things with the limbs to absorb all that energy, land on the surface without bouncing, and grip. And by the way, that's the main failure mechanism. If something goes wrong, what's likely to happen is that you'll just bounce off the wall and fall. So the question is, if we had instead an actively controllable suspension system on a small robot like this, how much better could we do? So the question then is, well, okay, how do we choose compliance? And there are a couple of different kinds of applications. We can think about small running robots or about the flying robots. And we've seen the value in tuning the suspension system, but things change. The environment may change. You may go from hard ground to sand, or the robot may change. You may change payload or something like that, or some details of the task may change. Well, this is not a new discovery. In fact, people have been working on tunable suspension systems for a number of years, and various approaches have been taken. One of the well-known ones is this idea of series elastic actuation, where you've got a sensor and you're measuring the compression or extension of this elastic element, and you're then doing a feedback control through the actuator to vary the apparent stiffness of the suspension. However, I would argue that this is not really directly changing the stiffness of the system. First of all, your ability to do this is limited by the bandwidth of this closed-loop control system. Also, you are, in terms of energy, paying a price for maintaining a certain apparent stiffness. In contrast, a whole nother class of systems does involve directly mechanically changing the stiffness of some mechanism. And so, for example, here we have one of the legs of the Rex robot and a motor and a rack and pinion system. So there's no energy cost associated with maintaining a particular stiffness, only with changing between stiffnesses. But as you can also kind of see, one of the issues with this whole class of systems is that they tend to be fairly complicated. It takes time to turn the motor and drive that changing spring system in and out. Some newer designs that come out that are more compact, but they're still quite limited in terms of the speed with which you can vary the characteristics of this system. And it would be certainly difficult for me to put something like that on an airplane where every gram is precious. There are also antagonistic systems. You can take a nonlinear system of actuators or springs, and by adjusting both of them together, you don't change the equilibrium position, but you do change the stiffness of the joint. So as we look at these various approaches,
when we're really thinking about small, fast robots, they all have some disadvantages. We're concerned about complexity. We want to be able to change performance very rapidly. That whole landing procedure takes about 1/3 of a second, so we need something quite a bit faster than that. So, electroactive polymers. These two have been around for, oh, well, at least a decade. They've been kind of in the category of a solution looking for a problem. They have some interesting muscle-like properties, and people have, there's been a fair bit of work on modeling them, looking at different configurations of stretchable films, diaphragms, bimorphs, and also some work on improving the manufacturing process, and it was even suggested, in fact, that they could be used as part of a variable suspension, although not much was done on that. Just to review briefly, and there was a talk just the other day on using electroactive polymers for a adjustable lens. The way they work is that you've got a capacitor with a stretchy dielectric material. So as you apply a voltage, you create a Maxwell pressure, and that draws the plates together. And then because this dielectric material is bulk incompressible, it has to stretch in the orthogonal direction. And note, by the way, that the pressure goes as the square of voltage over thickness. So you tend to want to use high voltages and have very thin films. It is a little bit unusual as an actuator because if you want to get work out of it, you want it to pull. That happens when you remove the voltage and let it relax. And that's just something to be aware of. So from a design point of view, what do we want? Well, we need an dielectric material that can withstand high voltages. We'd like it to have a high dielectric constant. More importantly, though, remember that V over T squared, it has to be very uniform thickness. And the main failure mode, actually, of these actuators is that you get a little bit of extra local thinning somewhere, and then immediately the field goes up and it fails right there. For various reasons, most people are using these 3M acrylic films. They're very tough, and they have very good uniform thickness. And it's interesting, when you strain them quite a bit, the dielectric constant goes up. The electrode, remember, this thing's stretched quite a bit, so we need an electrode material that is capable of withstanding a fair bit of strain. Again, most people are using some kind of carbon and grease or silicon oil mixture. And finally, as you recall, you have to pre-strain the material, and then when you remove the voltage, that's how it contracts, and you can get some work out of it. So that means you need a frame to hold it in, and this too should be a good insulating material and stiff and so on. We're using fiberglass, which we can cut on a laser cutter. So this is kind of where the state of the art is, and everybody who's worked with these electroactive polymers is well aware of the common problems that arise. The big one is that if you do have any little deviations from a perfect surface finish on your frame or any local flaws, you're going to get a little bit of local thinning in the film, and that's a disaster because as soon as you do that, the field goes up and you immediately get these arcs. And these sorts of pictures are all too common and well known for anybody working on EAPs. And you can see, by the way, empirically that the incidence of failures tends to be clustered at the inner and outer diameter. So, about a year or two ago, Sanjay put this slide together when talking to our lab group and said, why would anybody choose to work in robotics with a thin, very fragile, stretchy film that breaks easily? And we cover them with this nasty, goopy silicon oil with carbon in it that gets all over your fingers and onto your clothes and onto your lab bench. And then you energize this stuff at 6,000 volts, and you're going to get shocked sooner or later. So really, what is the motivation? And what this led us to was a fairly concerted effort to improve the manufacturing process, to improve reliability and improve yield. So the way we're doing it now, and this is for the simple reason that we needed to have enough of these devices that we could really do tests on them and start to put them in robots. What we're doing now is we're building them in a batch. We're using acrylic frames and with fairly precise alignment features. We use the same VHB film that most other people are using, and we use masks to successively spray on either the electrode or a anti-tear coating. And the basic design looks something like this. We've got the pre-strained dielectric. We'll do a batch of, say, 5x5 actuators at once. We, through a mask spray, a silicone rubber anti-tear feature. We put on the electrode. We put on the laser cut of fiberglass frame. And then you can actually stack multiple of these devices up in parallel if you want to get more force. So here, just in detail, this is the edge coating. This turns out to be really important. This both reduces stresses at the periphery of the film and also thickens the dielectric, thereby reducing the field, thereby reducing these many failures that tend to occur at the edge of the film. So as a result, we have gone, instead of sort of handcrafting these actuators that in a process that takes days and then it looks nice, but as soon as you hook it up to 6,000 volts, bzz, it's gone. We now have the build time down to about an hour. The yield is nearly 100%. They're quite reliable. This number, more than 10,000 cycles, I should point out that those are electrical cycle changes. If you hold it at 1 voltage, you can, of course, excite it. You can oscillate it many, many times mechanically. It's really the going from 0 to 6,000 volts and back that is an issue. And I think they can go many more than 10,000 cycles. It's just that's what we've tested it to. And what we're also finding is that all the actuators in a batch are very consistent. Okay, so let's talk about testing them. We're using a muscle lever, same muscle lever that biologists use for testing the muscles of animals. We can apply controlled forces or displacements and look at the resulting displacement or force to characterize them. And what you see is something like this. So first of all, a number of people have been modeling the properties of this VHB film. It's quite nonlinear. Of the various models, this one, the Ogden model, seems to fit the best. But the point I want to make is that for most of what we're doing, all this is that 400% pre-strain that we're doing, and we're mostly working up there. So as a result, it's actually reasonably linear in the range that we're working on. And I was thinking about Barry Trimmer's talk. Yes, when you exercise these things, there's a notable hysteresis loop. That may in fact be a good thing, although actually when we put it in a diaphragm, and the way we're using it, we don't have very much hysteresis, and again, it's fairly linear. So that means that we can fit these things. Remember, we're mostly interested in the dynamic response. So we can fit these things with the kind of standard viscoelastic model where you have a large number of basically springs and dampers in parallel, and you take out as many terms as you need to capture the behavior of this in a relaxation test. And in our case, to take it out to about 4 gives us a pretty good fit for the kinds of frequencies that we're interested in. Okay, so we're putting this in we want a variable suspension. So we're putting this, our current design has this as a diaphragm. And the way that it works is that we have this large pre-strain that we've applied. So you should think of it as being like a timpani or a kettle drum, that most of the stiffness that you feel when you press on the head of the drum is due to how much tension you've applied to the drum skin. And when we apply a voltage, what we're doing is we're relaxing some of that pre-tension. And so we get this interesting nonlinear response in terms of stiffness. And what, as I said, the voltage, applied voltage doing is mostly relaxing some of that pre-tension. And that's what gives us the change in stiffness. And it's a dramatic change. We're getting a several-fold change in the stiffness. So the dots here are empirical, and the plotted curves here are a linearized model. And you can see it works fairly well up to a few millimeters and then starts to deviate a bit. And so we can go from, say, 15 to about 100 newtons per meter for a single diaphragm, and then if you need more stiffness, you can play with, you can play with diameter. You can also start stacking these things in parallel. The other interesting thing is that because these things do have a bit of nonlinearity, if you want to sort of control really what your stiffness curve is, you can start putting them in parallel where you've pre-strained them different amounts in different directions. So if I make an assembly like this with three of these different diaphragms, and they have different curves, force versus displacement, when you add them up, the dotted line here, as you can see, the stiffness is actually fairly constant over some range of travel. And so there are games like that that you can play. The other thing to be aware of is because changing stiffness does not by itself result in much motion, the response time is very fast. So it's basically the amount of time required to charge and discharge a small capacitor on the order of milliseconds. And that means that it is suitable for things like our airplane application. So I think I went backwards. There we go. Okay, and by the way, although we need 6,000 volts, that doesn't mean we need a large power supply. You can get these very small kind of sugar cube-sized high voltage power supplies. The point is it's mostly a static charge, very low current. And so as long as you're willing to have a buffer capacitor, you can vary the voltage on something that you could even put on board a small plane. Okay, so it was nice to see Barry's work loop plots in the talk this morning. These are much simpler, but the interesting point is that muscle, of course, is not an electric motor. Depending upon how you vary stiffness as you go through a loop with force and displacement, you can either remove energy, you can add energy, or you can get some more complicated kinds of things that may be useful for other tasks. And so because we now do have a suspension whose properties we can vary very rapidly, we can play the same kinds of games. Yeah, once again, I'm going backwards. There we go. Okay, so just to sort of demonstrate this, we built a very simple 1 degree of freedom hopping robot. It's on a long arm with a pivot, and it's hopping up and down. Here you can see 2 diaphragms. We're hopping up and down. There's a load cell underneath. We're using a laser displacement sensor and a small IMU gyro to get velocity. And just as an example of what you can do, here are some of these. These are very simplified work loops compared to any of the ones that Barry showed. But you get the idea. If you just take the passive behavior of this, there's your diaphragm. We're letting it fall. As you can see, it's very underdamped. It takes quite a while to settle. Conversely, if I set this up with a braking work loop, and by the way, the dotted line is the actual response. It damps out much faster. Or conversely, here we have 2 diaphragms, and I can set it up to add energy, and I can achieve sustained hopping. All right, so where are we today? Well, let's go back to our intended application. This is what we're very interested in looking at. Remember, what we have is something like an airplane or a robot. Instead of thinking about a steady state application, for example, running or jogging, where you want to tune the performance of the system to adjust to hard or soft ground, if you're landing from a single jump, or if you're flying and landing on a wall, now you have a one-time transient event. And the question is whether you can do something interesting if you've got a system whose stiffness you can change rapidly enough that you can do it over the duration of that event. And as an example, this is a phase plot. It turns out with these airplanes perching, the most important of the state variables is pitch, so pitch and pitch rate. And what you can see here is that what basically happens is that in order to land, we're governed by a set of constraints that we can't violate. For example, on maximum force, on maximum displacement, we can't let the legs come in too far or we just crash. There's also constraints on things like the angle of the toes and so on. So if we have, in this case, a low stiffness, well, first of all, if we just start out here and we follow the dynamics, we're immediately violating one of the constraints. On the other hand, if we have a stiff system, we're fine, we follow the dynamics, but then at some point we're going to violate one of the constraints. In this case, it may be on, probably on maximum force on one of the spines. On the other hand, if we follow this trajectory, and then at point B, we now can switch from a stiff to a soft system. Now we follow a new trajectory and we land where we want to be. So this is the kind of thing that we want to explore with the airplanes. All right, so in conclusion, we assert that electroactive polymers are well suited for use as dynamically adjustable suspensions. This may, we think, be a better application than trying to use them as motors. They tend to last longer in this kind of application. Part of the reason being that you're just sort of augmenting their passive properties. They're already nice, sort of stretchy, somewhat damped suspension elements, and you're just sort of modifying that basic behavior. Having a batch manufacturing process where we can get the tolerances up has done a great deal to improve the yield and the durability of the devices. And we think a particularly interesting application is transient events, like landing at the end of a jump or a glide. Thank you.
Great, questions? Okay, there's Andreas in the corner.
Hi, very nice presentation, another very nice presentation. So there is some scaling laws that dictate electromechanical conversion, you know, and so it depends on the scale of the machine. You know, one would prefer to do electromagnetic versus capacity. I mean, whatever you have there,
right.
Electrostatic. So have you? I mean, I have no idea where this break point is. Where will it be for systems like this? Why not that inductor?
It's a very good question. Actually, the scaling works better at small scales. This is why when you look at MEMS devices, a lot of them are capacitive. So why aren't we going smaller? It's all about the manufacturing tolerances that we're able to achieve. In fact, the diaphragms that I showed you would work better if I could make smaller ones. And then, unfortunately, the alignment tolerances for these processes of spraying the electrode and the anti-tear coating and the other coating through a mask become that much more critical. Yeah.
Other questions? So now with the new system that you demonstrated, or that you have developed, you did show us the properties in phase space.
Right.
But how well does it actually really work in the real world? In what range, under what range of conditions did it really improve performance as compared to the first demo?
We're just now at the point of trying to put them on the airplane. What we know from the hopper is that, well, as I said, we anticipate that we're going to be getting about a 6 maybe fold variation in stiffness as possible. So what does that mean in terms of an airplane that wants to land? Is that a big deal or not so big a deal? I think that under ideal circumstances, you could kind of increase the envelope of, in state space, conditions under which you could land. Yeah, you could probably double it.
Okay.
Right now, right now we do a sort of very carefully controlled maneuver in order to get on the wall.
Yeah, because the other thing is that, like if you take the squirrel example, it's more like a parachute, right, approaching a surface.
Right.
Well, your platform is basically a winged aircraft.
It is.
So the flight dynamics is very different. You
Are
have
very
much more
different.
upwards force, for instance, when you hit that surface, as the squirrel would have.
That's right.
Right? So isn't it also biasing in some sense, the experimentation here?
For sure. And that, I mean, that's a whole other question. Why are we using airplanes? Well, because we wanted something that would fly a kilometer or two and then go land on a wall.
All right. You don't like these flying squirrels. They're not
They
efficient
won't
enough.
do a kilometer or two.
Exactly. Very good. Any other questions? Okay,
Yeah.
Stefano. You like running, no? Oh, your friend likes running more. Okay.
Yeah, I wonder whether, since very tiny carrots are involved, as you said, if there is some noise problem. I mean, the real world. If you have some,
noise problem?
from electrostatic charges that can build up in your devices. Yeah.
Well, it's a good question. And maybe kind of a prescient one. There's some other work we're doing on a hand with SRI, and they're using electrostatic brakes. And the control of the electrostatic brakes is something that we're having to be very careful about because we also have a bunch of tactile sensors on the fingers, and everything has to be really very carefully shielded. So certainly it will be something to think about.
Okay, great. Well, wonderful. Thank you,
Yep.
Mark.
All right.
All right, then our last presentation of the morning session is by Ricardo Zucca, who will be talking about cerebellar memory transfer and savings during motor learning, a robotic study.
Okay, so welcome. Thank you for accepting the paper to be presented here in the conference. I'm going to talk about a phenomena that is quite common observed. That so normally what we can see is that when we relearn some kind of motor skill, like for example riding a bike or playing a piano, whenever we relearn this activity later in time, we can see that normally this is a faster phenomena. This phenomena has been called saving in the context of classical conditioning, and it has been matter of different kind of studies from the behavioral and also the neural point of view. Just to make a brief introduction about what is classical conditioning. So I'm going to show some example about conditioning of the eye-blinking response. And actually what happens in classical conditioning is that when you pair a neutral stimulus that normally doesn't elicit any kind of response with a noxious stimulus that normally elicits a reflex response, at the beginning what we can observe is that we will have an eye closure at the presentation of the air path. But if we train the subject to pairing a lot of time the neutral stimulus and the US, what we will observe is that after a certain number of trials, we will obtain a conditional response, so an anticipated response that is predicting and in some way protecting the eye from the arrival of the unconditional stimulus. One characteristic of conditional responses is that they normally are well timed, so they are not elicited by the CS so they are well timed, so they peak just before the arrival of the US, and they are adaptive. So whenever we can change the delay between the CS and the US, what we will observe is that with more learning, the response will peak again just before the arrival of the US. After a session of training, what we will observe is this kind of learning curves. So acquisition will gradually, during the acquisition training, the subject will gradually acquire the conditional responses, while whenever we stop to give the US or we train the subject with unpaired CSs and USs, what we will observe is a gradual extinction of the response. The phenomena that we are also investigating with this study is that whenever you extinguish the response, if you give again a paired training of CS and US trials, we will observe a faster and more rapid reacquisition of the conditional response. This is observed for different kind of phenomena, and this is an example taken from training in eye-blinking conditioning. After more or less 40 years of study, starting from the preliminary ideas of David Marr and James Albus and the monography from Eccles and Masao Ito, now, after 40 years, there is quite a general idea that cerebellum is involved in this kind of learning. So, the first pioneeristic studies on looking at searching for the base of the learning mechanism in the cerebellum are from the 80s, from the group of McCormick. And actually, even if there is a lot of study, it's still not clear whether what's the role of the cerebellar cortex and of the deep nuclei there is still a contradiction in defining the precise role of these two structures in the cerebellum. One hypothesis is that the cerebellar cortex is responsible in defining the timing of the response, while the deep nuclei are responsible for the expression and the memory, so transferring a long-term memory of the response. Actually, the idea is that once the response is acquired in the cortex, it is slowly let's say, translated to the nuclei to retain a longer-term memory. In our group, we were developing in the context of the framework of the distributed adaptive control. We previously developed a cerebellar model, a neuronal network that mimic from the point of view of the anatomy and the behavior of the cerebellum. This model has been tested also on robotic devices, trying to learn an obstacle avoidance task. The main, the model is formed by different structures, so it's replicating the anatomical structure of the cerebellum. So it has a granular layer that is responsible for the delivery of the conditioning stimulus. A compartmental model of the Purkinje cells that is formed by the dendritic part of the Purkinje cells. One compartment that is inducing the spontaneous activity of the Purkinje cells and the soma of the Purkinje cells. The response is defined by the deep nuclei, and while the US is carried by the inferior, is sent by the inferior and carried to the Purkinje cell by the climbing fibers. The main idea of the model, it's based mostly on the Maralbus theory, so that there is depression, so that the paired presentation of CSs and USs induces depression of the synapse at the parallel fiber and Purkinje cells, driving to a pause in the Purkinje cells. Actually, this is the mechanism, the learning mechanism implemented in the model. So whenever we have the presentation of a CS and a US, at the beginning of the trial, what we will have is a spontaneous activity of the Purkinje cells, so that actually inhibits the deep nuclei and no response can be observed. At the end of the training, after a certain number of presentations of CS in the USs, what we will observe is that this synapse will be depressed, inducing a pause in the Purkinje cells, and that actually can release inhibition to the nuclei and perform a conditional response. The same kind of behavior has been observed also at the cellular level. So these are data taken from a paper of Yarmundeslo group. And as you can observe, this is the trace of a Purkinje cells during a training acquisition. So this is the first trials, and the cells is normally spiking, is spontaneously spiking at more or less 50 Hz. After more or less 350 trials, what we can observe is that the Purkinje cells start to develop a pause, and this pause is reflected at the behavioral level with a conditional response, so the closure of the eyelid to the presentation of the CS. During extinction, the phenomena is, let's say, reversed. So given an unpaired presentation of CS in the US or CSs alone, will induce a slow recovery of the activity in the Purkinje cells that in time will just go back to its spontaneous activity. So with the model that we developed within the group, so the main idea, as I told you, was that plasticity in the Purkinje cell, in the parallel fiber Purkinje cells, is the main phenomena that is required to learn the responses. And what we have shown is that actually this mechanism is sufficient to acquire well-timed adaptive responses. Another point that is important in this model is that it's based also on the idea that Purkinje cells, nuclei, and inferior olives form a negative feedback loop. In terms that whenever the Purkinje cell stops to fire, the release of inhibition to the nuclei will actually make the nuclei fire more and inhibit the inferior olives, so impeding the delivery of the US to the Purkinje cell. In this way, what happens is that the learning mechanism is entering an equilibrium. And so we can observe that, like here, that the learning stabilizes in time. The same experiment was repeated with the robot, and the robot was trained for a long period of time and to acquire the behavior, so to learn to avoid to collide with the wall of a circular arena. After the study, we decided to the model that was this model was intended to try to explain this idea that the timing is happening at the level of the cerebral cortex, but it didn't take into account other anatomical evidence that were showing that the connectivity between the pons and the direct connectivity between the pons and the nuclei. And so we extended the model in order to include also this kind of connectivity. And based on different studies, that in vitro studies that shown that there could be also potentiation and depression of this synapse between the mossy fibers and the deep nuclei, we decided to introduce also this mechanism of plasticity inside the model to see if it can explain this phenomena of savings. So the main idea of the studies from Pug and Zhang are that you can obtain potentiation in this synapse whenever you have a high frequency stimulation from the mossy fibers and at the same time a release of inhibition from the Purkinje cells. So this is the classical phenomena that happens during conditioning. On the other way around, you can have, it has been shown that we can have also depression whenever there is only high frequency stimulation of the mossy fiber to the deep nuclei. So we included these two kinds of plasticity into the model. The model actually is developed with IQR, that is a network simulator developed within our group, and is based on a generic type of integrated fire neurons. So we modeled these two kinds of plasticity. So at the cortical level, the LTD, so depression is induced whenever we have the concomitant presentation of climbing fiber stimulation and parallel fiber stimulation. On the other way around, we have a potentiation whenever there is only parallel fiber stimulation. In the nuclear site of plasticity, plasticity is defined as potentiation whenever we have rebound current, what we define as a rebound current, so this value delta, that actually is a release of inhibition from the Purkinje cells and activity in the mossy fibers. Depression is defined whenever we have only whenever we have activity in the mossy fiber and inhibition, strong inhibition from the Purkinje cells. Once we define the model, we decide to we try to do some to run some simulations with that try to approximate a conditioning protocol, a Pavlovian conditioning protocol. So we train the network with 10 trials, 100 trials of paired CS and US stimulations, followed by an extinction phase presenting only CS trials, and a subsequent reacquisition phase where we again presented CS and USs together. Actually, the kind of stimulation that we give, the CS is defined as a single pulse in the parallel fibers that is translated in a decaying trace in the dendrite of the Purkinje synapse, while the mossy fibers is driven by a prolonged pulse. The same for the climbing fibers. Climbing fibers are also defined. The US is defined by a single pulse that is released at the end of the CS presentation. So we are trying to reproduce a delayed conditioning paradigm, so where the CS and the US co-terminate. So as I show just the performance results of the model. So we replicated more or less a kind of learning curve that is observed behaviorally also with the model. So a gradual acquisition of the response that tend to reach an asymptotic level, and then it just fluctuates between 80 and 90% of responses. The mechanism that works during acquisition is that actually we have the parallel fiber to Purkinje cell synapse slowly depress until we have the appearance of the first conditional response. At the same time, plasticity in the nuclei start to be observed, and a partial memory of the association is transferred to the nuclei. Given the negative feedback loop, we have that the plasticity at parallel fiber and Purkinje cells stabilize, so that what we observe is that that's why we don't have 100% of learning, so the learning stabilizes around 9 trials out of 10 for each block. So after having the acquisition sessions, we just started an extinction session where we're just presenting the CS alone without the US. What we observed is that there was a quite fast decrease in the number of responses, and this is mainly due to the fact that parallel fiber to Purkinje cell synapse undergoes potentiations, and while the extinction mechanism is mostly driven by the potentiation of the parallel fiber to Purkinje cell synapse, while at the same time the synapse between the MOSI fiber and the deep nuclei still undergo potentiation during the first part of the trials, since until we have a conditional response during the training, we still have a bit of potentiation, and then slowly start to decrease whenever we don't have any more. So whenever the pause in the Purkinje cells is Purkinje cells undergo to spontaneous activity again. So as a third type of experiments, we try to reproduce reacquisition. And so we again introduce the paired presentation of CSs and USs, and what we actually had is let's say a not very strong effect, but there was an increase in so the first CRs started to appear faster than during normal acquisition. This is explained by the fact that plasticity in the nuclei was already high, so there was already the nuclei were already more excitable, and so what happens is that the learning in this case is just dependent on the LTD at parallel fiber and Purkinje cells. So there is no more necessity to have plasticity in the nuclei that is already formed, and so the first response can be observed faster than before. We then try to interface this model with the in a robotic environment. So what we did is try, even in this case, try to find a task that could approximate in some way the eye blink conditioning. So the very simplest model that we define is having a robot inside a circular arena, and with the task of trying to avoid the collision with the walls. So what we observe here is that the arena we used, so the robot was placed at the center, and this red patch was defining the CS occurrence, while the wall of the arena was the correspective of the unconditional stimulus. The robot is equipped with two kinds of sensors, so a camera that is the one that detects to detect the CS, and 4 frontal proximity sensors that are detecting the US. The robot is also equipped with a reflexive behavior that is that whenever it the four proximal sensors detect a contact with the wall, the robot just make a counterclockwise turns and start to go back into the arena. The robot is freely moving into the arena, so it was at first placed in the middle of the arena and just leave it there to move freely. So what happens is that in time, the robot has to learn the association between the occurrence of the red patch and the occurrence of the wall. So that in time, the robot has to learn just to turn just before reaching the wall. An interesting fact is that with this kind of setup, we are not controlling the timing of the occurrence of the CS in the US. So depending also on the direction of the robot, we could have different interception with the CS, and so different interstimulus intervals, so the time occurring between the interception of the CS and the occurrence of the US. So we ran a series of simulation, and what you observe is that the robot was actually able to learn the task. Gradually, there were less and less collisions with the walls. And so what happens is that the robot at the beginning is just driven by the UR behavior, so the reflexive behavior. And after, let's say, almost 20 trials, the robot starts to learn to avoid the collision with the wall. And the problem is that at the beginning, whenever we have plasticity, so the plasticity is still forming, there could be some conditional CRs that are conditional responses that are, let's say, later in time and doesn't allow to avoid the collision with the wall. So it takes a bit longer to have an effective behavior. After the behavior stabilizes, the time of occurrence of the CRs oscillate between different values, because what happens is that whenever the robot learns the response, and whenever the US is not occurring anymore, the LTP, so the synapse start to be potentiated, and this means that the pause in the Purkinje cells start to shorten, and so we will have a fluctuation also in time of the response. After having acquired the response, what we did is a conditioning, an extinction session. So what happens is that actually what we did is just shutting down the long-term depression in the model, in order to, because we wanted also to control whenever the US was coming, and so what we observe is that the robot gradually unlearned, if we can say unlearning the association, but in this case we still had more responses rather than in the simulation, because there were residual responses that were due to the longer ISIs given by the different time of interception in the arena. Given this, once the responses were extinguished, we retrained we switched again on the LTD in the model, and we started to observe if the model could learn faster responses, so it could maintain a memory of the association. What we observed is that the robot was able to perform faster responses, and they were also more stable in time, and also the timing remained more stable. This is explained because now the plasticity in the nuclei reached an asymptotic level, and what we observe is that the nuclei were more excitable than at the beginning, and the response were more, let's say, effective. So what we present here today is a model based on the anatomical and physiological properties of the cerebellum. The idea was to try to see if we could extend the model in order to make the model able to acquire, not only acquire a response, but also maintain an association of the memory, so that it can be reproduced later in time. The main phenomenal observation is that actually plasticity in the nuclei would facilitate the response of the nuclei, making the nuclei more excitable. So there was an increase in excitability of the nuclei that allows for this facilitation of the responses. One of the point that we want to underline here is that we use, this kind of mechanism are also used in different kind of models that are more abstract and on the other way around more detailed on the physiological point of view. But the idea here was that we try to have a model that could be as minimal as possible to make it able to work with the real world devices. So, and another point is that actually in the cerebellum there are different kind of, different areas are plastic. So there is not only plasticity in the cerebral cortex, but it has been observed in the last years that there are different kind of plasticity also at the level of the inhibitory interneurons or between the climbing fiber and the Golgi cells. So this was a proposal of a mechanism that can work, but still the results show that we didn't have a very strong effect of saving, and probably this effect is also mediated by other kind of plasticity that we are going to investigate in future work.
All right, questions for Ricardo. Nathan.
I suppose this question relates to your last point. So if I remember correctly, in eye-blink conditioning experiments, there's often a lot of pre-training where you don't get a response, but there's still learning taking place in the system. And then you get the acquisition trials that you see. So that means when you do the extinction, you're not taking the animal back to its naive state. So that could give an alternative explanation for savings. I was wondering how you could disassociate that from the mechanism in the deep nuclei that you're talking about.
So you mean that, well, actually there is, it's not really clear if the animal is going back to a naive state. So
exactly.
Yeah, so the idea is that, also what I base this idea is that there are studies that show that if you give longer, the longer the extinction that you give, the more the animal extinguishes the response, so the less observable is this saving phenomena. So there are these studies from Gormesano and Keogh where they show also from Molk in 2002 that shows that the longer the extinction, the less saving phenomena you can observe. So it seems that after giving extinction, what you observe is that the cerebral cortex is going back to a naive level, while there could be some sort of latent responses or some residual plasticity in the nuclei that can drive the saving.
How would you generalize your mechanism in second, third, and higher order conditioning, and what changes in the relearning effect?
It's something that at the moment we didn't think about. So, yeah. Yeah, that's true. Yeah. So that with the the idea, the main idea is that within the model we take this negative feedback loop between the Purkinje cells, the nuclei, and the inferior lobe, that is able also to explain phenomena like blocking, so that whenever you have you can use a second CS that is no more effective if you already trained the animal with so the idea is that the inhibition from the nuclei is the one that is driving the phenomena. So it can be generalized.
And in the larger DAC framework, what we're proposing is that basically you have an event-oriented loop and a time-oriented loop. And the cerebellum is your time loop, and it's in some sense completely agnostic about what an event is, right? So also if you talk about higher order conditioning, this can also be a redefinition of the fundamental events that are being piped through the pons into the cerebellum, right? So we see really as separated systems, but higher order effects are regulated in a competitive fashion in the cerebellum, as for instance shown in blocking, and how this is implemented in this negative feedback loop over the inferior olive. Okay. Of course, this can all be wrong, but that's the hypothesis. Other questions? All right, great. Ricardo, thank you. Some announcements. Yes. So like I mentioned yesterday over dinner, but you guys were all busy drinking, and some of you weren't there. So for the Convergent Science Network, this conference, Living Machines, was an experiment, because it's also a way to probe a field, to probe a community. Like, okay, is there critical mass in the community to really try to push something? And I think the overall impact, I think, and the responses we got to the conference are such that at least Tony and myself are very optimistic about the future of what we're trying to realize. And now this is in some sense also further assisted by the European Commission, because we know now that this network activity, the coordination action that we run, the Convergent Science Network, will continue for another 3 years. So this also gives us now resources and visibility to advance the conference. And so we want to do two things. So on the one hand, we think it's important we start to think also collectively about forming something you might want to call an association for the advancement of living machines, to also in that sense consolidate a community. So this is one thing we want to do. And if you have ideas about this, or you think it's a really bad idea, let us know. Then we can try to adjust the plans. This is one thing we're going to do. And the second thing is we are now already planning the follow-up conference, so our second Living Machine conference, which will take place now in London next year about the same time. It always rains in London, so you have to bring your boots and your umbrella. But okay, you know, it's going to be London. So these were the two announcements, and if you have ideas about this, if you, for instance, if you live in London and you want to help out with putting people up in your extra rooms or in your house or so, just let us know, because this is just the beginning of building a community to support and also drive research in biomimetics and biohybrid systems. So now, with that, I wish you a very pleasant break and try to find a nice lunch. Then we're back here at 3, where we have 2 further talks. They're really worth your effort, so come back. Don't eat too much, so that this brain stops working and you only have your gut being active. And after that, we have our prize ceremony, which will be worth your while as well. So we'll see you at 3 o'clock. Thank you. Huh? Oh yeah, and remember to vote, right, for best poster, because otherwise I'll take it home and you will not like that. Yeah? All right, great. Thank you.